on. So most of their lives, cells hang out in this limbo period called interphase, which means they're in between episodes of mitosis, mostly growing and working and doing all the stuff that makes them useful to us. During interphase, the long strings of DNA are loosely coiled and messy, like that dust bunny of dog fur and laundry lint under your bed. The other thing that happens is that all of the DNA begins to replicate itself too, giving the cell 2 copies of every strand of DNA. To brush up on how DNA replicates itself like this, check out this episode and then come on back. Now the cell enters the first phase, or the prophase, when that mess of chromatin condenses and coils up on itself to produce thick strands of DNA wrapped around proteins. Those, my friends, are your chromosomes. Instead of dust bunnies, the DNA is starting to look a little bit more like dreadlocks. And the duplicates that have been made don't just float around freely, they stay attached to the original, and together they look like little X's. These are called the chromatids, and one copy is the left leg and arm of the X, and the other copy is the right leg and arm. Where they meet in the middle is called the centromere. Just so you know, these X's are also called chromosomes, sometimes double chromosomes or double-stranded chromosomes, and when the chromatids separate, they're considered individual chromosomes too. Now, while the chromosomes are forming, the nuclear envelope gets out of the way by, completely disintegrating. And the centrosomes then peel away from the nucleus, start heading to opposite ends of the cell. As they go, they leave behind a wide trail of protein ropes called microtubules running from one centrosome to the other. During the metaphase, the chromosomes attach to those ropey microtubules right in the middle at their centromeres. After being pulled in this direction and that, the chromosomes line up right down the middle of the cell. Now, so far we've gone through the interphase, where the centrosomes and DNA replicate themselves and get ready for the split, the prophase, where the chromosomes form and the centrosomes start to spread apart, and the metaphase, where the chromosomes align in the middle of the cell. And now it's time to separate the chromosomes from their copies. This time, motor proteins start pulling so hard on the ropes that the X-shaped chromosomes split back into their individual single chromosomes. Once they're detached from each other, they're dragged toward either end of the cell. In the last phase, telophase, each of the new cell structures are reconstructed. First, the nuclear membrane reforms, and the nucleoli form within them, and the chromosomes relax back into chromatin. Then a little crease forms between the two new cells, which marks the beginning of the final split. That division between the two new cells is called cleavage, and all that's left is to make a clean break. This is done by cytokinesis, literally cell movement, by which the two new nuclei move apart from each other and the cells separate. We now have two new cells, each with a full set of 46 chromosomes. These clones are called the daughter cells of the original cell, and like identical twins, they are genetic copies of each other.